phosphodiester bond. Then this is into a secondary structure of double strand. Okay, so that is this primary structure. So two DNA strands come together and form a double helix structure. Then we have a 30 nanometer chromatin fibril kind of arrangement, which is further arranged into 30 nanometer chromatin fibril, and then which is further, went into supercoil. So these are the level of organization which we are going to see. We have already seen primary structure and double stranded structure. Now we are going to see the 10 nanometer chromatin fibril. Are you all clear? Why this packing of DNA, why this organization of DNA is very, very important? Okay. So let's look at, so we are now going to see the 10 nanometer chromatin fibril. But before that, we have to understand a very important packing protein that is called as histone proteins. So this is the most abundant chromatin protein, and it has almost 6 types of post-transcriptional modification. Okay? So these histone proteins are basically again classified into 2 types of histone protein. One is a core histone protein and one is a linker histone protein. So core histone protein is this octamer. Octamer means there is 4 here. So H2, H2A, H2B, H3 and H4. So these are core histone proteins. Okay, and so they will form an octamer. It's like a cylindrical structure, and on this core octamer, this core cylindrical structure, the double helix DNA is being wound. Are you clear? And this forms a 10 nanometer. Okay, 10 nanometer. So this entire thing is 10 nanometer. This is a 10 nanometer chromatography. So we have our alpha helix. Think of alpha helix as a rope. Okay, and then think as a cylinder, and on that cylinder, that rope is basically wound. Okay, so that cylinder is our histone octamer. Is it clear? Histone octamer. This is core histone proteins. Core histone proteins is H2A, H2B, H3 and H4. Is it clear? And this is the diameter of this is 10 nanometer, and that is why this is called as 10 nanometer fibril. Okay, so this is what you can see is as a 10 nanometer fibril. Okay. And various 10 nanometer 1, 2, 3, 4, 5, 6, 10 nanometer fibril are connected by this linker histone. Okay. This is linker histone proteins. So now what happens if you see? This becomes a 30 nanometer chromatin fibril or a solenoid. So we had alpha helix. That alpha helix is wound on the cylindrical structure, and 6 such cylindrical structures are further put into a, and this is what we call as 30 nanometer chromatin fibril. Okay. So what happens? Core histone protein, we have got H2A, H2B, okay, and H3 and H4. If we see H2A and H2B, these are dimers and arginine rich, and H3 and H4 are tetramer, they are lysine rich, and all of them form histone octamer. Very important. H2A and H2B are dimers, and H3 and H4 are tetramer. This is arginine rich, this is lysine rich. Linker histones are H1 histone, and which is seen in the linker region here. These are linker histones. These are loosely bound to nucleosomes. Okay. So this is what we have seen as 30 nanometer and 10 nanometer. So this, are you clear with the histone proteins? Okay. So now we will see the 10 nanometer chromatin fibril once again. So we see that double helix DNA is a 10 nanometer and 30 nanometer chromatin fibril. Okay, and this is how it is bound. Okay, it consists of nucleosomes separated by a linker DNA. Okay, this 10 nanometer. So this, are you clear with the histone proteins? Okay. So now we will see the 10 nanometer chromatofibril once again. So we see that double helix DNA is a 10 nanometer and 30 nanometer chromatofibril. Okay, and this is how it is bound. Okay, it consists of nucleosome separated by a linker DNA. Okay, and this is almost 1.75 times. This for each octamer, this DNA is bound 1.75 times. Okay, and it almost 1.75 times. Okay? So this is six such six cylindrical structure are bound together in this form. This is a 30 nanometer. Okay? This is group of nucleosomes formed from DNA and six such. This is one nucleosome. This is second, third, fourth, fifth, sixth. So six such form, and because it is six, this will be 30. Okay, now let us understand difference between euchromatin and heterochromatin. So we have seen the packing, how the packing has happened. So first double helix bound over core proteins, then six of such core proteins nucleosomes are linked by linker DNA, and one such becomes 30 nanometer chromatofibril. Now we have to understand the difference between heterochromatin and euchromatin. Very important. So what is heterochromatin? This is highly packed chromatin in the nucleus. What is euchromatin? This is loosely packed chromatin in the nucleus. This is generally inactive transcriptionally. It means they are packed form. And this is active. Why it is loosely packed? Because it is transcriptionally active. It contains more DNA. Obviously, it is loosely packed, so it will have less DNA. And this is less abundant and present only in eukaryotes. And this is 90% of human genome. This basically stains very easily and highly, and this stains very lightly. Okay, so euchromatin regions will be staining lightly. Okay, so heterochromatin are again of two types: constitutive and facultative. Constitutive are always active, and facultative are inducible. So constitutive always condensed, easily, essentially inactive, and these are sent in centromere and chromosomal end. And facultative, at times they are condensed, but at other types they are uncondensed and active. Transcription. The last level of organization, DNA organization, is supercoiling. Okay? So we have seen DNA primary structure, secondary structure, 10 nanometer chromatophore, 30 nanometer chromatophore together. Okay, so supercoiling is basically when they are further twisted together. That is called as supercoiling. So this structure is basically supercoiling. Okay? So this is in relaxed state, this is in supercoiled state. Under electron microscope, if you see DNA. So bDNA can be in relaxed superhelical linear B-shaped DNA. This is in supercoiled state. Under electron microscope, if you see DNA. So it is basically in relaxed superhelical linear V-shaped DNA, and this is thermodynamically more favored, but biological activity is reduced. So there can be two supercoils. One is positive supercoil, one is negative supercoil. If the direction of supercoil is in the direction of original rotation, then it is positive supercoil. And if the direction is opposite, so consider a rope which is alpha helix, and if we rotate it in the same direction of the original reagent, then it is positive supercoil. And if we rotate it in the opposite direction, then it is negative supercoil. What is the function of supercoiling? Again, it promotes packing of DNA into compact structures and facilitates DNA strand separation, replication, and recombination. So there is a very important enzyme, topoisomerase, which is associated with supercoiling. Okay, so this is nicking and resealing enzyme that can relax and insert supercoils, and because they can insert, this can nick, they can relieve torsional strain. Is bound to the, is it hanged? Okay. So basically, topoisomerase are the enzymes which are nicking and resealing enzymes. They can relax or insert supercoil, and they can. Can you see me now better? Okay. So this is relaxed state. This is positive supercoil state. This is negative supercoil state. And topoisomerases are basically what is responsible for supercoiling. So let's look super better. basically what is responsible for supercoiling. So let's look at some questions before we end.